to just sort of dive in, where we left off a long time ago, seemingly, was we had begun talking about the different macromolecules that build all living things, the different types of molecules that are found in each and every living thing, from bacteria all the way up into big living organisms like elephants, like you, kangaroos, lizards, etc., etc., etc. The list goes on and on. And the first macromolecule that we talked about was carbohydrates. And these end in OSE, so glucose, fructose, galactose, sucrose, maltose, etc. We're going to see lots of different carbohydrates. And we talked about how in all macromolecules, so carbohydrates, lipids, proteins, etc. In all those macromolecules, you can take each of those big classes of molecules and break them down into primary building blocks, things that can be sort of put together just like Legos can be put together with other types of Legos. And you can build bigger structures from those very small building blocks. And these, for carbohydrates, these are our small building blocks, and there's only 3 of them. So for carbohydrates, you can only build using 3 different types of Legos. You've got glucose, fructose, and galactose. And although they may look differently, so with glucose and galactose being these hexagons, so 6-sided carbon-oxygen rings, and fructose being this 5 or pentagon-sided carbon-oxygen ring, although they may all look different slightly, they all have the same chemical formula, which means that they have all 6 carbons, 12 hydrogens, and 6 oxygens. And this is all review as of now. And we looked at how when you want to make a disaccharide, or when you want to stick 2 of these building blocks together, such as in sucrose or table sugar, that's when you stick a glucose and a fructose together to make a disaccharide, a polymer. Sucrose is the example I was just saying, milk sugar, lactose, grain sugar, maltose. These are all disaccharides, or 2 building block polymers of sugar. And we talked about how instead of it being, like back here we said, each of these individually has 6 carbons, 12 hydrogens, and 6 oxygens, you would then think that it would be C12, H24, O12. But that's not the case. We actually have 2 hydrogens and an oxygen missing whenever we make a disaccharide, whether it's sucrose, lactose, or maltose. And the reason is because these are made using dehydration synthesis. And to do dehydration synthesis, excuse me, to synthesize a bigger thing by putting together 2 small things, or monosaccharides, you have to remove an H2O in order to do that. You have to remove a water from in between them. And when you do that, you subtract out 2 hydrogens and 1 oxygen. Again, this is review. And for our purposes, a change I like to make, instead of having, there's a lot of different nomenclature that you can use when you're going about counting how many links of monosaccharides are included in your carbohydrate chain. So a monosaccharide is just 1 of these, a disaccharide is 2 of them, and a trisaccharide would be 3, and then they get into oligosaccharide, whatever. There's a lot of different nomenclature that I'm not going to put you through. What I am going to do is say that anytime you have 3, such as we have here, or more carbohydrate building blocks bonded together in your polymer, such as in vegetable sugar or raffinose. Okay, just like we have here, we have glucose, we have galactose, glucose, and fructose bonded together in this polymer. Anytime you have 3 or more, you're now looking at a polysaccharide. So it goes monosaccharide, disaccharide for 1 and 2, and then if you have 3 or more, like we have here, you're looking at a polysaccharide. And raffinose, or vegetable sugar, is one example. And now this is something that you all hemmed and hawed over writing down because it was lots of words, but these are the 3 ways that carbohydrates get stored as polymers for future energy, and it depends on what type of organism you're looking at. For starters, over on the far left here, we have glycogen, and glycogen is only used as storage for future carbohydrate energy in animals. And so found in animals, we can digest it. If you were to all of a sudden start feeling a little bit cannibalistic and you ate a human being, you'd actually get a lot of future energy in the form of carbohydrates when you ate the liver, the muscles, etc., etc. and we'll get to glycogen in just a second, because as you can see, this is a molecule of glycogen. There's lots of ends here. And now that may not seem like a big deal, but what's interesting is that carbohydrates, whether it's glycogen or starch or cellulose, no matter which one it is, you can only ever break these down at the endpoints here. So boom, 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 boom. You can only have enzymes breaking down those long polymer chains into those digestible forms of monosaccharides. You can only do that at the endpoints here. And so glycogen breaks down the fastest, simply put, because it has more endpoints for enzymes to latch onto and thus break down. I was gonna cut out the fact that a dog just walked by, that two opposing dogs just walked by my house, and both of them were losing their minds, and it was actually really funny to watch. So maybe I'll include that into the final cut, but who knows, we'll see. Either way, we were talking about glycogen and how quickly it breaks down. This makes it really good for storing it in your muscles and in your liver for when you have that oh no type of scenario, like you see a bear in the woods and you gotta run away. Glycogen is a really good source of fuel to store because you can store it and then you can break it down quickly right when you need it, like when you encounter a bear and need to run away. And so this is why it's found in our muscles. Okay, that's why your muscles have it there, because your muscles are what need to do a lot of the oh no type of behavior. And so having that quick source of digestible energy right there is really helpful. And what's really interesting, if you're an athletically minded person, or you're just looking for sort of one-stop shopping when it comes to eating something that has both a lot of protein and carbs, is that shellfish, particularly things that basically the harder it is to crack open, so things like abalone, which I'm sure very few of you have actually had, but it is delicious. But basically, the more exotic the shellfish that you're eating, usually the higher the concentration of glycogen or carbohydrates that's found in that muscle, that animal's muscles. And what's really cool is that if you're an athlete and you want to cook one thing that has lots of protein for recovery and lots of glycogen to refuel your glycogen stores after a big bout of, some sort of athletic endeavor, shellfish is a good one-stop shop if you are looking to only cook one thing. And now we get on to starch. So now we're in the realm of plants for both starch and cellulose, but we'll start with starch because it's the simpler of the two. It's found in plants. We can digest this just like we can digest glycogen. We can digest starch, and it's the energy reserves in plants. This is what makes up things like fruits and seeds and other forms of products from plants. Since we can digest them, most of the food that we associate being starchy, like corn, potatoes, peas, etc., legumes, starches are a great source of energy. They are found in plants. We can digest them, and they make the energy reserves in plants. So this is a lot of times plants will store their energy reserves in things like seeds or fruits or vegetables. So potatoes, corn, peas, these are all great examples of starchy vegetables because plants have decided to hold some of their future energy in those products, both for them, but then we come along and pick them because we're hungry folk. And it's not as branched, if you look back at glycogen, it's not quite as branched as glycogen. It doesn't have quite as many ends for those enzymes to attach to and break down. So it digests quickly, but not quite as quickly as glycogen, which actually kind of makes sense because if you think about it, plants don't have very many, oh no, I need energy now moments. So it actually kind of makes a lot of sense that their method of storing energy for future endeavors would digest a little bit slower than glycogen. And now when we get to cellulose, this is actually only found in cell walls of plants. So we've gone over the different components of cells before and how they have their cell membrane, but then they're also enclosed in this tough outer layer, this cell wall made of cellulose, which is just one strand, singular strands of glucose linked together in long strands for future energy, not for future energy, but for constructing the cell walls and these tough fibers. We cannot digest this. Like I said, it forms the cell walls, and this is what gives plant its fiber. So this is when doctors are telling you to make sure you eat lots of fiber. It's telling you to eat lots of plant material, whether that's a fruit or a vegetable or something else. It's telling you to eat a lot of that because exactly why you can't digest it. Exactly because you can't digest it. And it's that fact that it can't be digested that it actually acts as sort of, I don't want to call it a snowplow, but having things moving through your digestive system that are undigestible, they tend to be a lot more robust in nature. And because they're so substantial, moving through your gut, not being digested, that actually ends up acting like a scraper, much like a snowplow for all the bad stuff that gets left behind in your gut. And so that's why there's a notable difference between somebody who has a high fiber diet and how clean their digestive tract gets cleaned out regularly versus somebody who doesn't eat plants or who doesn't eat plants at all. Their insides look a lot more like they need to be a good cleaning, et cetera, et cetera. And so that actually ties up our carbohydrate notes, and we finally get to move on to lipids, which are fats or hydrocarbons, mainly because they're made out of hydrogen and carbon and a little bit of oxygen here and there. Lipids as a class of molecule, their primary objective is to act as our protectant and our secondary source of energy. It's depending on which publication you're looking at, it's either said that lipids either have no true monomer, but I'm not sure that's entirely accurate because lipids do have some principal building blocks that we can look at. And we're going to look primarily at two. We're going to look at fatty acids and we're going to look at glycerol molecules, and that can vary moving forward. And so whenever we look at a monomer of a lipid, what we're really looking at are these fatty acids right down here. Now, what these fatty acid tails pair with is what ends up making the polymer. And so there are a couple different polymers that can be built with these fatty acids depending on what the fatty acids pair with in terms of other Legos. So we've been using the Lego analogy for some time now to describe how small building blocks like these fatty acids can build up to build bigger structures, just like you can take small pieces of Legos and build different things out of them. Now, I think if we were to keep this analogy of Lego pieces moving forward, what we'd have to then say is that sometimes you can use one type of Lego with other types of Legos to help build what you're trying to build. And that's just the case here. So depending on which of these, either a glycerol or a phosphate group, the fatty acid tails that are down here, depending on which one of these that the fatty acids pair with is what ends up building the different types of polymer or bigger structure you're looking to build. And so when you have these fatty acids that have this long hydrocarbon tail and this carboxylic acid group down here, what you end up having is a fatty acid that has two distinct ends. And having these two distinct ends is what helps it, A, bond to either the glycerol or phosphate group, but also give it some interesting properties when it comes to its chemistry. And so this end down here, the long hydrocarbon chain, which in these images is just this zigzaggy line, mainly because chemists are lazy when it comes to their nomenclature. At every junction here, there's a carbon with what's assumed to be the perfect amount of hydrogen, which is enough hydrogen to fulfill all four bonds of the carbon at this point right here. Oh, sorry, I clicked there. But that's neither here nor there. The long and short of it is that this right here and these right here are essentially the same molecule, just written out differently. But the long and short of it is that the long hydrocarbon chain is said to be, and I put these two words on the left side because the long hydrocarbon chain here and here are on the left side of the molecule. And they are what's called hydrophobic or water-fearing, right there, because they are nonpolar, or because this part of the molecule doesn't have a net charge. Meaning that if you were to just slice off this part of the molecule right here and you were to see if there was a charge on this end of the molecule versus this end of the molecule, you wouldn't find one. It would be totally, totally neutral. And because of that no net charge, that neutrality of that part of the molecule, what you end up with is a part of the molecule that doesn't like to dissolve in water and thus goes away from water and is water-fearing, and we call that hydrophobic. This is interesting because this is what gives you that oil on water effect, because oil, just like this, is just a hydrocarbon. Different types of oil, whether it's olive oil or butter or something like that. When you try to dissolve it in water, it won't dissolve. It goes away from it. It bubbles to the surface. It separates itself from water, and that's because of this chemical or characteristical portion of that part of the molecule. And if we wander on over to this side of the molecule, the right half of this molecule, this is called the carboxylic acid group, and this, like I said, is what helps it attach to either the glycerol or the phosphate group that we talked about in this back slide here. The other interesting thing that this carboxylic acid does is that it gives the molecule a totally separate set of chemical characteristics and physical characteristics. So if the left half of this molecule is hydrophobic or water-fearing because it doesn't have a charge from one end to the other, then this side of the molecule, the right side of the molecule, actually is the exact opposite. It's hydrophilic, which means it's water-loving, and that's because if you look at this little bit of molecule right here, different parts of this have different net charges, a plus on one side and a minus on the other. And because of that polar nature, that net charge from one end of the molecule to the other, you end up having water-like or water-loving characteristics, which we call hydrophilic, that cause this end of the molecule to readily dissolve and interact with water molecules, which is a really distinct chemical characteristic. And from there, depending on how those fatty acids pair with either a glycerol or a phosphate group, gives you different types of lipids. The first is fats. You traditionally find these in animals. However, they are found in, if I'm not mistaken in saying, every type of living organism, but primarily it's found both in plants as well as animals. You then have oils, which are traditionally liquid at room temperature, whereas fats are generally solid at room temperature, although, again, that can vary. And oils are traditionally most characteristically found in plants, as you can see from the examples here. If I'm not mistaken, that's peanut oil, olive oil, avocado oil, and I can't remember exactly what type of oil that is, but it appears to be some sort of tree nut. And last but not least is waxes, and this is a really interesting type of lipid structure that ends up giving waxes a very distinct chemical characteristics. They can be both solid or liquid at room temperature, depending on how they are structured. And like I said before, fats are traditionally solid at room temperature, found mostly in animals, and usually are associated with negative effects to your health. Whereas oils are sort of the opposite. They're traditionally associated with being found in plants, although are found in all living organisms, are generally liquid at room temperature, and are also generally associated with better health. And waxes can be found in any organism, can be both liquid or solid at room temperature, depending on how it's structured, and are sort of not unrelated to health, but we'll get to sort of where they come into your person in a little bit. But first we need to build our first lipid. We're going to make a triglyceride, and this is called a triglyceride because when you actually make a triglyceride, you're bonding fatty acids to a glycerol. In fact, you're bonding three, count them, 1, 2, 3 fatty acids to a glycerol. So it's a triglyceride. And to do this, to make this polymer out of these monomers here, the glycerol and the 3 fatty acids, to take these 4 monomers and bond them together into one big polymer, we use the exact same chemical process that we've been using this whole time. And that is, you guessed it, dehydration synthesis, where we remove a water molecule at each of these junctions, an H, an H, and an O, makes H2O, and so that's a water molecule that gets removed here, here, and here. And in your notes, what you're going to do is you're actually going to carry this through. So you're going to remove this water molecule at each of these junctions and just maybe circle it, show an arrow pointing out and say bye-bye or something like that. And then you're going to show how this carbon, because this O, H, and H are now removed in the form of water, they got yeeted, you're now going to connect this carbon to this oxygen with that remaining bond. And so you have bonded this whole structure using dehydration synthesis to this glycerol molecule, and you just do it 3 times, and then you end up with your glycerol molecule over here, and you can draw that over on that side of your note sheet. And now we get to some sort of generic functions, because lipids are very important, whether fad diets like to admit it or not. When you are looking at lipids in terms of health, you're looking at a couple of core functions that you need lipids in your diet for. First off, your cell membrane. All of the cells inside your body separate the inside of the cell from the outside world. It's actually one of the most principal requirements for being a living thing, is having a definitive border between where you begin and where the environment begins. And that is for almost all of your cells. In fact, all of your cells, is the cell membrane. And this is made almost entirely out of lipids, what we call phospholipids, which is where you pair 2 of those fatty acids with 1 phosphate group, whereas a triglyceride, like you built over here, is pairing 3 fatty acids to a glycerol molecule. A phospholipid is just a phosphate group paired with 2 fatty acids. The other core function of lipids and fatty acids in your body are hormones, and these are lipids that are made into steroids to send chemical messages into our body. And we're going to get to how steroids are kind of unique, or steroidal structure is kind of unique, in just a second, because it pairs very well with the next portion, which is cholesterol, which is something most of you have heard in a negative context. Cholesterol is bad, blah, blah, blah. Well, if you didn't have it, when it got a little bit warm, a little bit warmer than about 100 degrees, you would melt. You would literally melt into a puddle of you. And what's awesome is that in cold temperatures, like we tend to have in New England, you would also freeze and become sort of solid and not really able to move well. Cholesterol is this unique molecule, again, that I'll show you in 1 second, that grabs on to all the fatty acids that are wedged into your cell membrane and holds them at exactly the right distance apart, so that you maintain this sort of solid fluidity, this sort of in-between zone where you're not melted and you're not frozen, you're just nice and jelly-like. That way you're not melting or freezing and you can stay optimum. And here's that cell membrane with all these dark yellow, or light yellow, bright yellow we'll call it, little tails that come off of these tan phosphate groups. Those are the fatty acids, the bright yellow portions right there, coming off of those tan phosphate groups. So that's a phospholipid with its 2 fatty acids coming out into the middle of this phospholipid bilayer, or your cell membrane. And these really bright yellow portions right here, these are your cholesterol molecules, which chemically are wedged all over your cell membrane and attach themselves to fatty acids, to different fatty acids of different phosphate molecules, and wedge themselves in there so that they're holding these phospholipids the exact perfect space apart, because if it were to get cold, they would want to crowd together, and when molecules crowd together, they solidify once it gets cold enough. And when it gets hot, they want to separate, because they've got so much kinetic or thermal energy heating them up and wanting them to get all excited and separate out. And what that does in that case is it grabs onto them and instead holds them closer than they would otherwise be. And I don't know if you can tell, but you can see that these have a very unique structure right here. And because of that unique structure, that bodes well into moving into steroidal structure, because although these are different molecules, which I'll get to in just 1 sec, what's really neat is that these all have steroidal structure, that waxy type of structure that not only is the structure found in our hormones, but is also the structure found in other types of waxes, such as cholesterol. Cholesterol is that lipid product that's really important for keeping that cell membrane doing what it's supposed to. And cholesterol has this steroidal structure, as do hormones, as do other waxes. And so here's an example of how just a very small chemical change, and I mean so small that this oxygen loses a bond to this main structure right here, and instead picks up a hydrogen. And that one change sends into effect a cascade of changes that makes either estrogen or testosterone. It's a very subtle change, but that very subtle change ends up making chemicals that are only slightly different. And as this one literally just has an aromatic ring as opposed to an extra carbon bond over here. And those subtle differences are enough to make this a chemical that promotes female types of traits in living in human organisms, whereas this is a chemical that tends to promote male types of traits. Although it absolutely and scientifically needs to be said that there are probably thousands of different factors that contribute to whether or not you develop traditional male or traditional female types of both physical, emotional, and cognitive traits. And there's a lot of gray space that exists in between these two chemicals. And last but not least, when you attach the idea of lipids to health in general, you end up with a common term called atherosclerosis, which is the hardening and narrowing of the arteries due to our body's reaction to high cholesterol or saturated fat levels inside your bloodstream. So here we have a normal human artery. And let's just pretend they went on a McDonald's binger for months and months on end. What would actually end up happening is lots of saturated fat, which is the type of fat that you find primarily in animals, as well as lots of sodium or other excess cholesterol coming from animals in that food. What happens when you digest food is the byproducts get pushed out to your bloodstream. And so when you have all that, you know, not great for you stuff floating around in this blood vessel, it actually ends up sticking a little bit to the endothelium, or the innermost lining of epithelial cells of your arteries. When that happens, your body actually sort of freaks out and sends a bunch of lipids, calcium, and cell debris to sort of break up the that little bit of sticky stuff that's stuck onto that endothelium. And it's actually your body's reaction to the products getting stuck to the endothelium that lead to this sort of fibrous cap, or this big, what we call a clot, forming. And so the long and short of it is that it's not necessarily the diet that ends up killing you. It's your body's reaction to the bad things in your blood that end up wreaking real havoc inside your arteries if your diet is poor. That being said, if your diet is not poor, this tends to not happen, although there are some noted genetic linkages where this happens almost regardless of your diet. However, those cases are much more few and far between. And the last thing I want to hit for the health connections is that when you go to the doctor, if you were to do a blood profile and you were to get your blood tested for a number of different things, they're going to run a lipid profile on you. And when they do this, what they're really looking for are two things: the levels of your HDLs and the levels of your LDLs. And they're going to go over that with you, and they might dive into it a little bit, but they also might not. And so what is an HDL? What is an LDL? Your HDLs are your high-density lipoproteins, and these are essentially a very unique molecule that float around in your blood that is sort of this combination between fat, lipo, and proteins. And your HDLs are high density, which means that in that combination of that big molecule, your lipoprotein, you have more protein in general than you have fat, meaning that protein weighs more or has more mass than fat when you compare the same volume of the two things. And so if you have more protein than you do fat in that molecule, you're going to have a high density or a higher density molecule. And it's often said that you want to keep your HDLs high because what's interesting is that when you have this molecule that's more dense, because protein is more dense than lipid, and you have more proteins than lipids bound together in that big molecule, when you have that big dense molecule, it tends to not stick around in your blood vessels. Because it's high density, it tends to move with the flow of your blood a lot better. And this is generally, these types of lipoproteins are generally associated from the fats from plants. And so this is, again, another tout for why plants in your diet are an absolute necessity. And the other level they're going to go over with you is your low-density lipoproteins, which for the exact same reasons has the opposite effect. And because proteins are more dense than lipids, In this case, if you have a low-density lipoprotein, you have more lipids than proteins in that big lipoprotein molecule. And what this causes is that almost like a raft effect, in that they end up floating a little bit easier in your bloodstream and thus come into better contact with those endothelial cells along the insides of your arteries and blood vessels and capillaries, etc., and end up sticking around in your bloodstream, in your arteries and veins a lot longer, a lot more severely. And these types of lipoproteins are associated with fats from animals. And so if you were to answer this question of overall, which is better for you, I think it's pretty clear you'd have to go with HDLs, which tend to come from plants. And this is sort of a very brief overview. We're going to hit the actual structure of this very hard when it comes to genetics, and for now we're just going to treat it as another molecule. For our purposes, RNA and DNA are the only molecules that are made of nucleic acids, are also sometimes called polynucleotides. And they have a very characteristic double helix structure. So if you were to take one thing and put it into a helix structure, this is just two helices or two helixes that are bound together in sort of the same format. And we're going to look more into that in just a second. The DNA molecule was discovered technically by Watson and Crick. They were the first to formally publish it. However, if you were to look in the literature, there's a strong string of evidence that suggests they stole very, very suggestive work from this lady Rosalind Franklin, who was genuinely one of the better X-ray spectrophotographers of her time. She was the one who actually did the work to figure out and surmise out what the structure of DNA looked like, and actually had some work published about it. And they didn't necessarily give her as much credit as they should have, and are not very well liked in the scientific community for that purpose. Either way, it needs to be said that Rosalind Franklin, pictured down here, likely did in fact die over the work that she was producing, because X-ray crystallography was the process she was using to basically crystallize bits of DNA, blast X-rays down onto them, and see the refractive patterns of those X-rays using a photochromatic paper. And at the time, they didn't have refined X-ray equipment, and so she was spending hours and hours and hours underneath this unrestricted X-ray beam that likely played a role in her death. That was incredibly premature due to cancer. And now when we look at the actual molecules of RNA and DNA, both of which are involved in the process of storing and moving information in order to make proteins, which is our last stop, our true last stop in the macromolecule chain. RNA stands for ribonucleic acid, and DNA stands for deoxyribonucleic acid. And so this is the main library of information that's essential for almost all living organisms, all known living organisms. And so what we have here is, again, a lot of what we go over here is very general and will be expanded upon when we hit genetics. But right here, this central line here, DNA getting transcribed into RNA, which gets translated into proteins, this is the most important concept we will go over in this whole class. This is called the central dogma of biology. But either way, DNA getting transcribed into RNA, which then gets translated into proteins, is the most important process that goes on in your cell, because proteins are what do and build almost everything in your cells, tissues, organs, organ systems, and thus in organisms. And the process for taking information bound up in your DNA and getting it through into what it needs to be to do what it needs to do for you as an organism is an unbelievably important process. I can't overstate that enough. And so when we actually look at these DNA and RNA as a molecule, we see that they are made up of nucleotides, which is the monomer of nucleic acids. And this nucleotide, or this monomer, this individual building block or Lego piece, contains a couple different components. It contains a sugar, a phosphate, and a nitrogen base. And that sugar and phosphate are sort of arranged into the backbone or the sides of a ladder, whereas the nitrogen base makes up sort of the middle rungs of a ladder that you actually put your hands and feet on in terms of the structure. And so here we have our sugar, phosphate, nitrogen base rung. And so if you were to imagine a ladder, you can imagine that if a bunch of these nucleotides are stacked on top of each other, one after another, sugar, phosphate, nitrogen, phosphate, sugar, nitrogen, and so you have a long string of vertical chains of this, you're going to have the sugar and the phosphate being the side rail of the ladder, and on the other side you'll have the nitrogen base being the rung of the ladder, but we'll look at that again in a second. For these nucleotides, you end up with different types of nitrogenous bases in the middle, being the rungs of that ladder. And so different nitrogen bases means that you have 4 different types of nucleotide. You have adenine, thymine, cytosine, and guanine. And if this were RNA, you would have, instead of thymine, just a uracil, which is a slightly different molecule. And so if you were to look at what the purines are, if you were to look at these different types of molecules as different chemicals, you end up with purines and pyrimidines. So A's and G's are the purines, and C's, T's, and U's, or cytosine, thymine, and uracil, are the pyrimidines. And you can tell that just based off of their structure. This has a double ring structure, it's a purine. This has a single ring structure, this is a pyrimidine. And what you find is that there's complementary base pairing between the purines and the pyrimidines. And so the purine, in this case, the A to T bonding, is the purine bonding with the pyrimidine. Remember, A bonds with T. And in this case, it's the purine over here bonding with the pyrimidine here. Purine G bonding with pyrimidine C. And then in RNA, the T is simply is basically swapped with a U, whereas the C and G remain the same. And this is a really important concept right here, is that the A's always bond with the T's and the T's always bond with the A's. And in RNA, it's just U instead of T. So in RNA, it would just be A to U and U to A. And G's always bond with C's and vice versa. C's always bond with G's, or cytosine always bonds with guanine. And this is something you have to commit to memory. I'm not a big fan of promoting just rote memorization for the sake of it, but knowing that A always bonds with T and vice versa in DNA, and C always bonds with G and vice versa, and knowing that you will never see an A bonded to a C unless there's some sort of drastic mutation. T will never bond with G or C, A will never bond with C or G, vice versa. You'll never have vertical bonding up over in that way. You'll only ever have A's bonding with T's and vice versa, and C's bonding with G's and vice versa. You must commit that to memory. And this is getting into what I was talking about, that ladder structure. Well, we're going to start over on the right and move left. But that ladder structure sees that phosphate and sugar, phosphate and sugar, phosphate and sugar making up the backbone of this ladder. And here, if I circle it right here, is our full nucleotide where you have the phosphate, the sugar bonded to that nitrogenous base. And in this case, we have a purine, adenine, which is bonded to its pyrimidine, thymine, on the other side. And so when you take this nucleotide and you bond it vertically, you end up with one end of the double helical structure of DNA. And so you have one nucleotide here, another nucleotide here, another nucleotide here, and so on and so forth going down. And if you go in the opposite way, you have that opposite helix going up and forming this bigger structure that I'll get to over here. But when you play this is what it looks like chemically. We're going to focus more on these types of structures over here, just because this is a little bit more complex than we're going to dive into. But what you have here is that you end up running the opposite sequence depending on which strand you're looking at. So if we start looking at this strand, which runs what's called from the 3' to 5' direction. And you run that in the opposite way, you end up just with the opposite code. And so if you had AGT on the left side here running, or sorry, if you had AGT running from the 3' to 5' right here, what you would actually have on the opposite side is the opposite sequence. So you would have, instead of AGT, you have TCA, which is that opposite sequence. And when you play this out into a bigger structure, you end up not with a straight ladder with these side rails that are made up of that sugar phosphate backbone here. You end up instead with the side of not a straight ladder, but a twisted ladder, where those side rails, that blue ribbon, is still that sugar phosphate backbone with those different nitrogenous bases which pair in the purine to pyrimidine way, that A to T and T to A versus G to C and C to G way. And you can just see that played out in these different colors going up the ladder. And that actually brings us, that's it for our nucleic acid section, and that brings us to proteins, which is our genuine last stop in our macromolecule train. Proteins, like I said before, make up the structures and the sort of workers or the enzymes of life. And like I just mentioned, enzymes are the little worker bees that end up speeding up all the internal chemical reactions that must happen for life to be able to take hold. And so whether we're looking at enzymes or we're just looking at structural proteins, we're going to be looking at molecules that have a new element in them, an N, a nitrogen, as opposed to fats and carbohydrates, which only had a C and an H. It does need to be mentioned that nucleic acids also have a nitrogen inside their structure. You can see it here in those nitrogenous bases, as well as a phosphate in the backbone there. But we will dive more into nucleic acids when we get to genetics. For now, with proteins, we know that the monomer, or the little Lego piece, the building block of that bigger structure, is called an amino acid. And it does need to be mentioned that there are 20 different amino acids in all living things. What's neat is that 11 of these 20 we can literally just make ourselves based off of other materials that we intake. Whether it's getting different materials from carbohydrates or lipids that we might intake or eat, we can make 11 of the different amino acids just based off of other materials. However, 9 of the different building blocks that make up proteins are known as essential because we cannot make them. We must eat those 9 amino acids. And that's why they're called essential, because they're essential for your diet. But we'll get to that in a second. When we look at the structure of an amino acid, the structure is remarkably similar for most of the molecule. This is called a carboxylic acid group, or what's shorthand called a carboxyl group. And this is called an amino group. And this is actually why we call it an amino acid, because this is a carboxylic acid group. So you have your amino group and your carboxylic acid group. Thus, it's called an amino acid. To the north, we have this lone hydrogen atom, which stays there no matter which of the 20 different amino acids they are. And this down here, this R side chain, is sort of like X in a mathematical equation. You can make that be whatever it needs to be. And this is the only spot where amino acids, the 20 different amino acids, are different from each other. Depending on what you slot in here, you can have one amino acid, let's say glycine, versus another amino. If you slot something different in there, you'll get a different amino acid, let's say arginine. Regardless of which type of amino acid you're looking at, if we just sort of bond 2 amino acids together, whether it's 2 of the same ones or 2 different ones, you end up with the same thing. We call that a dipeptide. Now, there are some specific names for dipeptides, but we won't dive that deep. Here, we have just a regular amino acid. We're just going to call it a generic amino acid, where instead of describing what slots into that sort of wild card position, we're just going to leave it as its variable to say that something could slot in here. But when we want to make a dipeptide, or if we want to link lots of amino acids together to make a big structure, so take our monomers and turn it into a polymer, we're going to use the exact same process that we've been using, and that is dehydration synthesis, where you take a hydrogen from the amino side and you remove it, and then you take an alcohol or an OH group from the carboxylic acid side, and when you remove that hydrogen, and by removing this water molecule, it now allows this carbon to bond straight to this nitrogen, which we call a peptide bond. And since we've now made a peptide bond between two amino acids, we now have a dipeptide. So there's a lot of different nomenclature that could go along with this. If you had 3 of these amino acids linked together by peptide bond, you could call it a tripeptide, etc., etc., etc. But we're going to cut it off at 2 just to make things simple for us. So you'll only ever hear me refer to a amino acid, which is a lone amino acid, a dipeptide, which is sort of a 2 amino acid combined together, and then a polypeptide, which will be chains of 10 or more amino acids. So if you have a polypeptide, then you have amino acids that are strung out at least 10 long. So you've got one long string of 10 or more amino acids bonded together with peptide bonds. You do not have a protein yet. If you want to have a protein, you have to have more than one chain of polypeptides. So you can have as long of a chain of polypeptides as you want, but that's not a protein. It's only when you have multiple chains interacting with each other that you end up actually having a protein. Proteins are very complicated molecules. And what's responsible for making those long polypeptide chains are ribosomes. And they take amino acids and they actually do this linkage of the different amino acids making peptide bonds. And they do this by dehydration synthesis in order to form multiple different polypeptide chains that end up becoming the full-blown proteins. And when you actually look at these proteins, you have multiple different levels of structure that you can see basically by zooming in on them. And so primary is our first step. And if you were to just have a straight polypeptide chain of amino acids, you're looking at a polypeptide, which is just primary protein structure. Now, the reality is that this primary protein structure actually ends up folding up into secondary protein structure, which is that straight chain ends up getting folded and clumped and twisted into a bigger structure, which we call secondary structure, which you can see here. And so these straight chains of polypeptides actually end up either going into 2 basic shapes, which is a pleated sheet, which is like a zigzaggy sheet, where you can imagine this being the sort of twine that ends up building this bigger sheet of material, either in a pleated form, a zigzaggy form, or this alpha helices form. And that brings us actually to third or tertiary structure, which is where this secondary structure which is where you can see those pleated sheets and those alpha helices have actually started to form this larger, I'm going to try and trace it all out here, bigger structure, bigger folding structure. And so if you're looking at just the alpha helix or the pleated sheets, you're zoomed in this far and you can only see that level of structure, you're only at secondary structure. But if you zoom out further and you see how those pleated sheets and alpha helices end up making this bigger network of shapes, now you're looking at tertiary protein structure. This is still just one polypeptide chain. This actually isn't a protein yet. And so you have to zoom out even further to see how this big network of twisting and folding interacts with another polypeptides network of twisted and folding. And now that you see multiple polypeptides in their big twisted folded structure, now you're looking at quaternary protein structure. You're actually looking at a full-blown protein here. And that actually is the end of our macromolecules. Those are the different types of molecules, their structures, their functions that build a lot of the chemicals that go on in your body. Our next step is going to be enzymes for next week. But that is to come. And so for now, that's all the information that's fair game for your macromolecule quiz. And I hope that this lecture helps. Study those notes and also go through those practice sessions in order to help you out. See you soon. Bye.